Hey everyone, welcome back to the channel. Today we're diving into a key topic in immunology and rheumatology, complement C3 and C4. These two proteins are power players in the immune system and crucial when diagnosing autoimmune and inflammatory diseases. So if you're a med student, resident, or clinician brushing up your knowledge, you're in the right place. Let's break it down. The complement system is part of our innate immune response. It consists of more than 30 proteins that work together to opsonize pathogens, recruit immune cells, and cause direct cell lysis. It can be activated through 3 different pathways. One classical pathway, triggered by antigen-antibody complexes. 2 lectin pathway, initiated by mannose-binding lectin binding to microbes. 3 alternative pathway, activated directly on microbial surfaces. And all 3 of these pathways converge at one major protein, complement C3. So let's talk about C3. C3 is made in the liver and is cleaved into 2 parts during activation. C3a, a pro-inflammatory molecule known as an anaphylotoxin. C3b, an opsonin that marks pathogens for destruction. Because C3 is involved in both the classical and the alternative pathways, changes in its levels can reflect general complement activity. So what causes low C3? Consumption, like in lupus nephritis, post-infectious glomerulonephritis, or membranoproliferative GN. Or genetic deficiency, which is rare but leads to recurrent infections. On the flip side, elevated C3 is usually non-specific. It might just be part of an inflammatory state or metabolic syndrome. Now let's look at C4. C4 is also made in the liver and primarily plays a role in the classical and lectin pathways. It doesn't participate in the alternative pathway. When C4 is activated, it splits into another anaphylotoxin, C4b, which helps form the classical C3 convertase. Low C4 is often a sign of classical pathway activation and is especially important in diseases like systemic lupus erythematosus, hereditary cryoglobulinemia, normal or high C4. That usually means complement isn't actively being consumed. Okay, here's where it gets really useful. Let's talk patterns. C3, C4, what it suggests. Low classical pathway activation. Think SLE or immune complex disease. Low normal alternative pathway activation like post-infectious GN or C3 glomerulopathy. Normal low classical only consumption. Hereditary angioedema or cryoglobulinemia. Normal. Probably not complement-mediated disease. These patterns help narrow down differentials when you're working up glomerulonephritis or autoimmune conditions. Let's apply this to real-world conditions. In SLE, both C3 and C4 can drop, especially with active nephritis. Post-infectious GN, you'll see low C3 with normal or slightly low C4. C3 glomerulopathy, you'll see persistently low C3 but C4 stays normal. In hereditary angioedema, low C4 is a key clue, especially with a C1 esterase inhibitor issue. And with cryoglobulinemia, C4 is usually low while C3 might be near normal. So when should you order C3 and C4? When you're evaluating suspected autoimmune disease like lupus, when working up unexplained proteinuria or glomerular disease, or when someone presents with angioedema, vasculitis, or other immune-mediated symptoms. But here's the catch. Both C3 and C4 are also acute phase reactants, meaning they can go up during inflammation. So context is everything. To sum it up, C3 is a central complement component, altered in both classical and alternative pathway diseases. C4 is more specific to classical pathway activity. Together, they offer valuable diagnostic clues in autoimmune disease, glomerulonephritis, vasculitis, and angioedema. So next time you're reviewing labs, don't overlook these 2 powerhouse proteins. If you found this helpful, give it a thumbs up, share with your classmates or colleagues, and subscribe for more breakdowns like this. Drop a comment if you'd like us to cover CH50, AH50, or complement deficiencies next. Thanks for watching. See you in the next one.